What makes a journey a discovery? Well, it starts with a first step, an often unlikely beginning, driven by a force that makes us question our world. Understanding that there is something to be found in everything, in everyone, that around us are little universes waiting to be unlocked if we look a little closer, question a little more, and try a little harder. And that the questions of doubt are the fuel that force us to continue, to struggle on. The risks are the reasons, and new possibilities are inspiration. The journey is greater than ourselves. And that when we finally look back at the unlikely heroes, the outliers, the disruptors, we understand that it is the people who question, who look a little closer, that are the ones that drive innovation. Celebrating the spirit of innovation.
Ladies and gentlemen, please welcome Dr. Ross Cagan for his talk, Conquering Cancer, One Fruit Fly at a Time.
Thank you. You can hear me, yes? I'm Ross Cagan, as you may have gathered. I am the fruit fly guy. And this is part of a fruit fly. Actually, that's the whole thing. What you're seeing here is actually the beating heart of a fruit fly, just to remind you guys that they really do have things that we do. Actually, my lab studies the heart as part of our diabetes project, but I'm not going to talk about that. What I am going to talk about is I'm going to focus on a question that my lab and I took up. It's been almost a decade ago now. We really jumped from a very different world. And we jumped into the world of disease, especially cancer. And I'm going to talk about how we make complex drugs, how we make complex models, and how we treat patients using my favorite model, fruit flies. And if I can convince you that that's a good idea, I probably have done some pretty remarkable things just by that. So cancer. Cancer is something that has been with us for really since we have been humans, and in fact, of course, other animals suffer from this as well. This is a fundamental part of a developing body, okay? At some point, you have to stop things from growing, and sometimes that stops working. 3,000 BC, 5,000 years ago, are some of the earliest reports of what are clearly breast tumors, where they tried to cauterize them, and it puts a note at the bottom saying, this is not treatable, okay? Things haven't changed that much. The way I like to think about it is that there are, if you will, 4 revolutions in cancer treatment. The first revolution is surgery. And we can see this here. An example, this was back in the 17th century. We're basically just taking out the primary tumor. This actually works, all right? The problem is when cells leave the primary tumor, then you get in trouble. And actually, over 90% of all cancer deaths come from metastasis, so that didn't work so well, okay? But that's still standard of care. The second revolution is more recent, chemotherapy. Let's throw things that are so toxic that anything that tries to divide will kill it, all right? And that actually still is standard of care for most cancers. And it works. But again, tumors generally find a way around it. The third revolution, which came about 20 years ago, and some of the people who are my age remember when this started, the idea was what was called targeted therapy. You were going to figure out what caused cancer, and you're going to drug it. That seemed like a reasonable idea. And here we are today. Cancer is now, as of, I believe this year, the major killer of Americans and major killer of, in the Western world. So it was heart disease, and I think there's something to think about with that. So I'm originally a basic researcher who really tried to understand how epithelia assembled, and we worked on the nervous system, all of this. And here's something that I learned. In basic research, mechanism is king. That is, if you get mechanism, you win, okay? But in applied research, which I really didn't know a lot about when we jumped into this, if you have a mechanism and you don't have therapeutic, you lose. And if you have a therapeutic and you have no idea what the mechanism is, win, okay? So for example, my dad, 17 years ago as of last week, had quadruple bypass surgery. And you know what? He's rocking. He just came back from a vacation in Alaska. He's doing great. Now, what causes heart disease? Unclear. Everybody fights about it. Doesn't matter. You can bypass it, and now it is no longer the major killer of Americans. Okay? So these are all sorts of lessons that we thought about. So let's talk about the fourth revolution in cancer with that in mind. And that is the era of personalized medicine. So the idea in personalized medicine is that you, for example, sequence a patient's tumor. So cancer is fundamentally a genetic disease. So if we just get a list of all the mutations, then we should know enough about the cancer to treat it. And so the problem is, I think it's a great idea and I'm all in. The problem is what we do today is we're still stuck in the third revolution, which is you sequence the tumor, you look in there and you say, hey, I recognize that mutation. Other people have that mutation. And I have a drug that hits that protein. Let's use that, right? So that's not really personalized medicine. That's kind of genomics by voting, right? If I can find enough people that has that, it must be important, and let's go ahead and drug that. Or as I like to put it to my science friends and really annoy them, hey, I studied that since I was a postdoc. It must be important for cancer. Let's drug that. So we're trying to get away from this, all right? And the way we're trying to get at it is to use my beloved fruit flies, all right? Because in fruit flies, you can do things that you can't do in anything else. They're the simplest model organism that actually has an epithelium, and therefore you can study solid tumors. Sorry, worm people. So fruit flies. Probably most of you guys here know a little bit about fruit flies, right? They're the ones where you cross the red eyes to the white eyes, and then somehow in that weird Punnett square, 2 generations later, it's that one, okay? So let me tell you what you can do with flies today, all right? And I'll just cut to the chase. You can turn any set of genes on or off in any cell, forget cell type, any cell at any stage you want, including adult, all right? And that power, we have leveraged that to try to rethink what you can do to create cancer models. You guys with me? So we're trying to reset the mindset of just because my model system can't do that, it must not be important. All right? So as you can tell, I love flies. Okay? All right, so how did we get into this? Well, let's start with one lonely graduate student in my lab, Renee Reed, really picked up this baton. The poor woman, I should say poor, she's now a professor at Emory. She picked up the baton and basically started to work on something that a neighbor of mine was studying. So I have to admit, when we started this, I didn't know anything about cancer. So let me just tell you, you only need to know 2 things about medullary thyroid carcinoma. One, it's a thyroid cancer and one of the few that will actually kill you because it commonly goes metastatic. And then you end up chasing the tumor by trying to pull it out of various parts of the body until it hits something important. Okay? And the second thing you need to know, and here's a high-resolution crystallographic structure of the thing that drives most of these tumors, which is the RET oncogene. So RET is, for those of you who study signaling, it's a classic receptor tyrosine kinase. It binds ligands like GDNF, it dimerizes, it activates, I don't know if I'm doing something to cause this, so maybe whatever I'm doing, I'll stop doing it. And basically, it signals through most of the pathways that we all know and love. RAS, SARC, I'll get back to them, okay? So how do you make a fly model of a thyroid tumor? So let me tell you guys right off the bat, flies don't have a thyroid. Now, they have a very skinny neck. We may have missed it, but I don't think so because they don't make calcitonin, thyroxine, any of the sorts of things that that you would want to see. So instead, what we did is we stuck it in the only tissue we were actually studying at the time, which is the eye. Okay? And I hope you can see this nice, beautiful patterned eye here. If you put, in this case, actually a human oncogene, or a human or fly, they both work the same. You stick it in the eye and you express it there. I hope you can see that it basically, you get a big mess here. You see little tumors coming out of here. Basically, if you pop the lid here and look underneath, you'll see switches in cell fate, proliferation. Cells will actually leave that eye and go around the body, okay, and form other tumors. We'll get back to that. all the things that you know, that we all know about in cancer, most of them we see here. This goes back to the, what do you do next? Now, if I was a true fly person, we would study the mechanism of this and try to understand, and we did do that. But this is probably the more important thing we did. Thinking about what the purpose of this was, is we developed a method to grow flies in a 96-well plate format. That's a little demeaning to the flies, but it works, okay? And as you can see here, we use a deep 96-well plate. We use robotics that were not created for this, so we had to tinker for a while. And we move fly food into each well. We move drugs into each well, and it mixes it up and down, and we have 4 different chemical libraries in the lab. We use a modified FACS sorter from Union Biometrica, where we actually essentially FACS sort 10 embryos into each well, so everything is automated. Okay, how cool is that? And then once we've filled everything, we put an oxygen-permeable lid on it. The flies hatch out. They eat the food. They eat the drug. We ask a very simple question. Can we find a drug that will make that tumor go away without killing the fly? Okay, you guys with it? Because I'm not going to explain that again. And we're doing this over and over and over again, okay? So just wave magic wand, drugs fall out. Actually, it's a little harder than that. All right, so obviously I'm telling you that it worked, and it actually worked better than I could have expected. So we did a screen. We had all sorts of advice which we should screen and this and that. We screened everything. This was back when I was at WashU Medical School. We screened everything we could find, basically, and we got exactly 1 hit. Excuse me. Will this work better? Okay. Okay. Is this better? Okay. Now I feel like a game show host. All right, so we did a screen and we got 1 hit. So let's look at this hit, okay? Now you can see here's our bad eye, our tumor eye, and then in the presence of this drug, ZD6474, the fly, we just fed it orally in the food, and I hope you can see that the eye is all better in the presence of the drug, right? So, and what was cool about this is that the eye was all better at a concentration 30-fold lower than was required to kill the fly or harm it in any way we could find. So if we were to call this a therapeutic index, and if we were oncologists, which I'm not, we would say that has a therapeutic index of 30. That's a ratio of helping to killing. And a typical cancer drug has a therapeutic index of usually about 3, okay? So I was very excited. I went running around the WashU Medical School saying, you should just give this to people. This will completely cure them. So unfortunately, it turns out you can't actually take drugs from flies and give them directly to people, which is annoying. But to cut to the chase, because I actually have other things I want to tell you, it turns out this drug had actually been in clinical trials 10 years before, which we didn't know initially. It went into, it was not made as a RET inhibitor or anything like that. It was for breast cancer. It was supposed to hit something called VEGF receptor. And the bottom line is it went into phase 1. It didn't help anybody. None of those drugs did. It didn't harm anybody, but it was shelved. And we essentially ended up inadvertently pulling it back off the shelf. And actually, the company that made it, AstraZeneca, was on our original paper. And they took it into clinical trial based on this and also based on data from Massimo Santoro, who I want to give credit, in Naples. It's fantastic having people we work with in Naples, because I love Naples now. And Sam Wells, who really was the clinician who drove this into the clinics. And to cut to the chase, this drug was approved in 2012 as the first standard of care for medullary thyroid carcinoma. It's still standard of care, okay? So that worked great. That worked out better than I expected, honestly. So what did I do? I moved to New York, and I turned the lab over, and we just switched to working on disease, mainly cancer, okay? Let's see if we can give this a full-on try. Let's talk about drugs. Okay. So now what I'm going to do, I'm going to explain in very quick terms, but you're all going to become fly pharmacologists in about 2 minutes flat. So this is work done by Dirta Das, an amazing postdoc in my lab, in collaboration with Cagan Shokot, probably the best academic medicinal chemist on the planet from UCSF, and his former postdoc, now a professor at Sinai, because we sucked him up. And what Dirta did is he set up a system where he expressed RET in several places in the fly. Half of them survived to pupation, but none made it to the adult. And we turned the transgenes on right before they pupated. Now, if I was a drug company, I would just say this is an easy problem. Let's just give them a RET inhibitor. Everything wrong with that fly is due to RET. How hard is that? But here's the problem, and this is, I think, thinking that has not been challenged, which is the idea that understanding what causes a tumor is the same thing as finding the best targets. That, I think, is not always true. Let's think about it. If you mutate something so that you just change one amino acid and now the cell freaks out, you're going to probably need that protein for other things in your body as well. And RET is no exception. So several RET inhibitors are put into trials. They basically all failed because they make you sick. For example, they'll denervate your gut. Okay? You can have real problems. And you can see that here in these flies, the highest dose we can give them before we kill everything, it does nothing. So now I'm just going to cut to the chase. We looked at a library that these guys made, and we got one hit. I love one hit, which I'll call AD1 for Arvindar1. And hey, look at this. Now we finally have some adults, and you can see it's not very toxic. They make it all the way to pupation. This is good. Okay, it's a good start. Now what do you do? So what we did was, after thinking about this, we basically fed the flies the drug. The flies have 270 kinases in their kinome, and we just knocked them all down one by one, all right? And just asked, in a whole animal system, can we improve this drug by figuring out what it should hit better and what it should leave alone? What we call balanced polypharmacology. So I just summarized a whole bunch of work. Let's see how it works. If we remove a copy of ERK, part of the RAS signaling pathway, look at this, all right? That says that we want this drug to hit ERK and its pathway harder. You guys with it? We call that a target. On the other hand, we identify what we call anti-targets. For example, TOR. If we remove a genomic copy of TOR in the presence of the drug becomes highly toxic, all right? So we just stepped through the kinome and went back to Cagan and Arvin and said, hey, can you make a drug that hits these harder and leaves these alone? And amazingly enough, they can actually do this. And I would explain to you how if I understood it, but I don't. But they send back two drugs, which I'll call AD1B and AD1C. And check these out. This is the drug we want. Not only does it allow them to survive to adult, it's extremely non-toxic. All right? And I'll just show you very quickly a little bit of mammalian data. So here is a thyroid cell line, an MTC cell line. Vandetanib, which is what ZD6474 is now called, works beautifully. But you can see AD1, and especially AD1B, work way better. But that doesn't impress me because if you throw bleach on here, it works even better. So what we did is we grew up tumors. We really grew them up till 46 days, really big tumors. And when we do that, vandetanib in a mouse has a hard time dealing with these tumors. So it's human cells implanted in a mouse. But you can see AD1B is much better. And importantly and critically, you can give AD1B extremely high doses. And it's extremely non-toxic. So this is just feeding mice these drugs. Vandetanib, you get lower body weight, but not AD1B. Okay? And by many criteria, these are fantastic drugs. And here's what we learned from all this. Okay? When we asked, with these drugs that we're making, and I'll show you one more example quickly. When we asked, not just what targets do they hit, but where they hit them in the animal, that's where the interesting stuff comes in. So these drugs don't just act on the tumor. Some of their targets are in the tumor, but some of them are elsewhere, in the skeletal muscle, in the liver, in the brain. These are things you can map out using a fly. And what we learned from that is, unlike what I call tumor-focused screening, whole animal screening gets you a different class of drugs that acts on the whole body. And these drugs can be terrific. Okay? And we can talk more about that later in the session. And so, for example, we're taking well-known drugs, such as sorafenib, which is a long-approved cancer drug. We've run it through the same thing, and we're making what we call super sorafenib, super trametinib. Okay? And these are things that are extremely non-toxic and work much better. And it's just by rebalancing the pharmacology. Okay? So I'm going to tell you two more very short stories. I promise very short. And then let's move on. Okay. So you guys with me so far? So you see the thinking. Okay? So now I'm going to flip this around. So complexity in drugs. Let's talk about complexity in models. All right? So colorectal cancer. 3% of drugs that go into clinical trials are approved for colorectal cancer, the #2 cancer killer of Americans. In fact, cancer as a disease has the lowest success rate of any major disease for clinical trials. It's the worst. Okay? Why would that be? It succeeds on all our cancer models. So should we conclude from this? Maybe we need some other cancer models to help out. Okay? So let's look at flies. So what Erdem Bangi, a really spectacular postdoc in my lab, what he did, because he was interested in colorectal cancer, in my lab it's pretty much one postdoc, one cancer. He went into the database and he asked, what's the most common combination of mutations that you see in these cancers? Okay? Just add them up. So for example, the most common quadruple combination you'll see in colorectal cancer is RAS, APC, P10, P53. He built that, aimed it at the fly gut, specifically a short part of the fly gut, and put all four in and only turned it on in the adult, okay, using magic fly tricks. Incidentally, that for those of you who are counting, that's 9 transgenes. Okay? He built the quadruple. He also built for the quadruple, he built 3 other quadruples, so he built 4 total. He built for each one all the triple subcombinations, all the doubles, and all the singles. All right? He really built everything and used that to take this apart. So what did we learn? First of all, let's take a look at some, because I keep showing the data. So let's look at some pictures, okay? So first of all, what he saw is overproliferation, multi-layering. Here's a portion of the adult fly hindgut. It's GFP is on here, green fluorescent protein, so we can see the gut glowing, and you can see polyp-like structures emerging from here, okay? Also, he sees multi-layering, distant migration of cells. I'm going to show you a picture in a second, senescence, apoptosis. A lot of the things that we see in cancer we see here. So for example, here's a portion of the fly gut, okay? Here's a muscle wall around it. Here's what's called a trachea, it carries oxygen. And what happens is, so cells are proliferating in this gut, things are getting bad. Cells are periodically extending a process through the muscle wall. When they find a source of oxygen, they'll corkscrew around it like this, and then the cell will walk out of the gut, hop onto the trachea, walk off to distant sites, and form secondary tumors and eventually kill the fly. So from beginning to end is about 3 weeks. Okay? And they remodel trachea into them, just like neoangiogenesis, all the sorts of things that you see. It's really amazing. All right, so why am I going through this? First of all, I can tell you it's really complicated what they're doing. Genes that do one thing alone do completely different things. And if you want to take quick notes, I'll hesitate for one moment here. Okay, that's enough. But the more important question, remember we talked about this, is what about therapeutics? What's the difference between a one-hit model, which is what the mouse models are, KRAS or the so-called MIN mouse, APC mouse, that all those drugs rotated through before they failed in clinics? What's the difference between a one-hit and a 4-hit? So Erdem looked at this. He took 16 drugs, many of which failed in colorectal trials, and he gave them to our one-hit flies. 13 of the 16 worked beautifully to knock the tumor out. But in the quadruple, none of them worked. That's why they're failing, okay? And because he has all, so you with that so far? So complexity really matters. And so remember, he has all the subset subcombinations, and so he was able to map out for each of these drugs where the resistance emerges. And using that information, he's created a drug cocktail that works by a new mechanism, which I don't have time to get into, a 2-drug cocktail that's able to handle this complexity by attacking several points. Okay, it's a very low specificity cocktail. Okay? And could we have ever dreamed up this cocktail? No, we just screen. There's something to be said for just empirical screening. Okay? And then we make up a story after we find what works. Okay, so I'm going to finish by talking about the effort that ties these together, and that's our patient treatment. This is just mouse stuff. Let's forget that. Mice. I want to talk about the Center for Personalized Cancer Therapeutics, which is really my newest baby, and which I'm very excited about. These are the fly guys who are part of it, okay? Peter, Jess, Erdem, and Alex, but just to be clear, there's probably over 30 people involved with this center. We have a Wednesday morning meeting that's amazing. Oncologists, clinicians, genetic counselors, bioinformaticists, fly geneticists, mouse geneticists, and so on, all show up. And we've really been building the structure from the ground up. These are my co-directors, Marshall Posner, who among many other things is head of clinical trials, and Eric Schott, who among many things is director of the Institute for Genomics and Multiscale Biology and has really been driving the genomics of this. Okay? So here's the question we're going after. How much complexity matters? And what we're starting with is we're putting in everything. And then we'll dial back after we see if everything works. So this is what we do. We go after 3 cancer types: medullary thyroid carcinoma, which I talked about, colorectal cancer, which I talked about, and triple-negative breast cancer. Okay? So tumor comes out, and pathologists at Mount Sinai, led by Michael Donovan and Nina Longtine, they make sure that it has high enough purity, at least 40%, which would be useful. If it does, then some of that tumor goes into a mouse, so-called patient-derived xenograft, and we put this aside. We'll get back to that mouse. The rest of it, and I hope you like this little graphic on sequencing here. The rest of it gets sequenced. Yeah, check this out. This is from Eric Schatz's group. So it's sequenced a whole exome, all of it, and we go through all the genes that are mutated. Okay? We decide which of the mutations actually alter protein function, which actually takes a fair bit of work. And then to cut to the chase, all of them go into flies. And Peter, in particular, Peter Smybert, has developed technology to rapidly do this in sort of patient time. And once we have that fly built, which we call a personalized fly avatar, you guys know exactly what we do. We have a library of 1,200 FDA-approved drugs. The FDA won't allow us to just give random compounds to people. Again, annoying, but probably a good idea. So to create these novel compounds, we create cocktails of FDA-approved drugs by screening through libraries through multiple times. Okay, so we pull out the robotics. Here we go. All right. Okay, and we screen through multiple times to try to identify 2 to 3 drug cocktails. And once we identify those cocktails through massive screening, we then have committees that vet the science of it, they vet the pharmacology of it. Dosing, as you can imagine, is actually tricky. If we have a successful mouse, we'll treat that. But probably more importantly, to get dosing and make sure it's not extremely toxic, we also work with Exemplar to give these drug cocktails to pigs for typically 10 days. And we monitor metabolites and so on. If everything looks good, then we work with the oncologist to propose a treatment. And I should say the ethos in our center is that the information goes out real time to the patient and the oncologist. A lot of physicians are really worried about that. They worried we would raise their hopes, but I find it's just the opposite. When you treat people with respect and show them the data, they're actually much more realistic about it. Okay? So this is just an example of some screening we did and created a 12-hit model. And I'm just going to finish by saying, if you look at that list of 12 hits for this particular patient who had medullary thyroid carcinoma, probably 2/3 of those, if you're an oncologist, you would never even heard of. Okay? Interestingly, this patient was also diabetic, and as I'm showing here, if you create a tumor in a normal fly, it looks bad, but if you just flip them onto a high sugar diet, it looks really bad and you start getting metastasis. That matters, and it changes drug response. So for that patient, we put his model on a high sugar diet as well. So with that, I am going to end. Thank you very much.
Well, welcome, Ross. Welcome again. Delighted to have you here with us. And as I mentioned to you, we're live, so we have mics. And we have also a student, Cagan. Where is Cagan? She's going to read some questions from the web that... which mic is Ross going to use? The same?
I assume I'll use the same?
Yeah.
Okay.
Okay. So, why are you at Mount Sinai? We talked a little bit about it. Let's start with that.
Sure. As one of my colleagues at UCSF said, that's either the most brilliant or the dumbest idea I ever heard of. And you have to take a leap. Okay?
Yeah.
Sinai is, although it's an old hospital, it's a very, much like Northeastern actually, it's a very young medical school and a very young graduate school. And they have the ethos of, you should be surfing out on the edge. And very few places have this, where there's a culture of risk-taking. And I know because I've asked many of my friends at many of the major, even cancer places, and they all said the same thing. Gee, I'd love to try that, but they would never let me do that. Sinai's attitude was, that sounds complicated, that sounds great. Let's figure out how to get it to work. And we got approval from the FDA and so on. It's just a matter of, taking risks. So the answer to your question is there's a culture of risk-taking there, which is why I moved there.
And in terms of funding, the traditional funding pipelines, NIH, etc., as you explained, they are not risk takers. So if you can elaborate on what you mentioned to me before, but also what kind of funding you're getting now.
Sure. And as we talked about before, I actually started not as a scientist, but as a musician. And my sense of scientists was that they're always surfing out on that edge, right, of risk-taking and of science. They're like professional risk-takers, right? Unfortunately, as many of my colleagues will verify, I very often get back but that might not work, which I thought was the point of why we were doing. If I knew it would work, I wouldn't actually have to do it. So the NIH really didn't know what to do with us. So basically, we are really from funding from the dean, who has been great, and probably soon from Philanthropy. This is the sort of thing that the NIH is just not set up to do. And what's ironic, it's other scientists making that call. It's not the NIH. So there has to be a change in culture there. There's no doubt about it.
And what about the company, the Medros? What does it do? Why did you feel the need to do it?
Yeah, Medros, which stands for Medicines from Drosophila, not Medicines from Moss. And my partner, Tom Baranski, who's a clinician who's been great. So what I learned was, again, the purpose here is to get treatments to patients. And what I learned early on, remember, I didn't know anything more about this than anybody here, most people here, which is, and this was a few years ago, which is how to start a company, why you would want to start a company. When we did the thyroid thing, that was for a rare cancer, and that rules were for what's called an orphan disease. And so we could actually move that pretty easily into clinical trials, because otherwise drug companies aren't going to care about them. There's only a few thousand patients a year that have this. For major diseases, once I saw that and I wanted to go after the big boys, what I found is, especially at the time, you couldn't discover a drug in your lab. Is it me?
I think it's you.
I think it's me.
No, don't worry about it. That's great.
What was I saying? Oh yeah, so for major diseases, there's a deep problem in bringing it from your lab to the patient, which is intellectual property issues. Basically, pharma has to put it through clinical trials. It's very expensive. I'm summarizing this. You know this. However, if the intellectual property is not clean, that is, that they can't get all of it, they've got more drugs in their own pipeline than they can deal with. Why would they care about you? Even if you swear up and down it's a great idea. So I talked to a number of people, and they said, well, you just have to start a company, and then if it works, the big pharma just takes your company away from you, and then they can put it in. So that's why we started the company. I think things are opening up a little bit more now, mainly because big pharma is starting to rely more on us to do the basic research that they used to do themselves. But still, at the end of the day, you want to put a drug and help people out, you have to start a company. And if I were to say anything, I notice there's a lot of young people here. I'm all about new generation biologists learning how to take risks. Our generation hates risk. We like tenure, we like grants, we don't like risk. And I'm worried that filters down to you guys. Similar to the computer age in the early 80s, where the government stopped increasing the funding and everybody went out and started companies. I urge you guys to think about that. There's a huge world out there. I love bio and biotechnology. There's a huge world out there if you're willing to take that risk about going ahead and starting your own little medroses. It's a ton of fun, and it's a great way to push this enterprise forward.
I'll have some opportunities to go back and ask you other questions, but Cayman, let's start with you. You have the mic already, correct? It will be on. It will be on. Hello?
Hello? Okay.
Yes.
All right, so this is a question from Twitter. It says, millions of people worldwide are battling cancer. Is this approach scalable?
That is a sharp question, yeah. So the question is it scalable? The answer is exactly the way we're doing it, patient by patient by patient. It is scalable up to a point, and then I think we're going to bump into reality in terms of we couldn't treat millions. So what we're doing now is we're building what we call an avatar army. So for example, for colorectal cancer, there's currently about 300 patients sequence that has been posted mainly by the TCGA, and then we're of course getting more patients. So we're making individual avatar lines from all of them. We're screening all of them with drugs to create drug cocktails from all of them. And then we're going for what we call best fit. You come in, you get sequence that's going to become standard, send that sequence to us, we'll do best fit. I don't know if that'll be as good as personalized, but for some patients that'll have to do. And then obviously the more flies we get, the better that database will be.
Ross, in this respect, you mentioned that you have opportunities now that you're considering to open other centers in other countries. Will this allow you also to scale it up too?
Yeah, so exactly. So obviously, as we expand outward into the rest of the world, so there's been quite a bit of interest in what we've been doing elsewhere. That will help bring in more patients, which of course will increase that avatar and will just give us more experience. And it has sort of put the crack of the whip to us to make sure we industrialize it in a way that can be exported elsewhere, instead of just having these brilliant guys doing it.
Cagan, would you like to go for another question and then we open it up?
Yeah. So I kind of feel like you already just answered this question, but I think it's still important. This one's from Facebook, and it's, do you envision a future where any individual can have their own personalized fly models, where they can test a variety of remedies for improved physical health?
Naturally, the answer would be yes. And so maybe riffing off of that, what I would say is we're not just doing cancer. We're also doing a number of other diseases, including RASopathies, a lot of rare diseases. We've looked into, we're working on diabetes. We just submitted something on diabetic nephropathy, which is kidney failure and so on. So the answer to your question is, flies will not be good for everything. There's some things they're missing. Their immune system's a little odd, that sort of thing. So you want to pick wisely, but with that in mind, I think they can be used for a number of different diseases. And so the answer is yes.
Thank you.
Thank you. Misha, do we have a question here from this side? If you have a question, raise your hand, we'll get the mic ready for you. Misha, the rule is to have a short question and a long answer. So you choose either the answer or the question.
You mentioned word oxygen 3 times. And you know why I'm counting, right?
I know exactly why you're asking about oxygen, Misha.
And
Explain to us, Misha.
We have a professional exchange. I miss his emails that I can meet him at 3 p.m. I want him to answer for everybody. Why in your last email you said if HIV regulates angiogenesis, in simple words? Why angiogenesis is important in your model? Because this is something that we can do together and improve screening in more physiological condition. This is very short.
Okay, that was pretty short. And thank you for the question, Misha.
Could you provide a context for the question
Yeah, of course.
too?
Of course. Maybe if I keep it not too close to me. So the question is, I think, is basically what is the relationship of angiogenesis and oxygen to tumors? Why would you want to go after it? And what have we seen in our tumor models that address this point? Did I get that? If not, I'm going to answer that one anyway. So the answer is, as I mentioned, one of the things you see in tumors is that when you have a secondary tumor, it needs oxygen, and so it sends out signals that are through a transcription factor called HIF, other growth factors, and it sort of rewires your arterioles into it, so it can get some oxygen, which is pretty evil if you think about it. And so there was an idea that if you could cut off the oxygen, you could cut off the tumor. Mm-hmm. And the difficulty was, and actually that first drug I showed you, ZD6474, was an example of that, is that when you cut off the arterioles, it started to grow up your arterioles and became more aggressive and didn't work out and then all got dumped. So to answer your question, we find that if you cut down HIF and stop the tumor from doing that you can stop the tumor cold. There's no doubt about it. And in flies, of course, we can do this genetically, so we can do it cleanly.
Misha, are you satisfied with that? You have a rebuttal. No, but you need a microphone.
I speak
No, because it's for the web.
Okay, so I completely agree. And after you suggested after your talk, I will describe to you, you have my paper already, how to use oxygen, supplemental oxygen to synergize with your drugs in human patients.
Well, we'll have to test it. Sometimes humans are not a good model for fruit flies, so we'll see.
Yeah, that's good. Any question this side? Yes. We need a microphone here.
I know you said that you're concerned about innovation coming out of the NIH, but clearly your work depended on things like the Human Genome Project in order to succeed. And we all, of course, are concerned about funding for research at the federal level. Can you talk about the importance of that as well?
Sure. You've actually put your finger on a root cause. And I hope I wasn't too harsh about scientists, especially because I am one. Basically, as funding tightens, people become more risk averse because I've got 3 proposals. One I know is going to work. Maybe it's not amazing, but I know it's going to work. And I've got 2 more. I've got to pick one of them, okay? So I can obsess over what's wrong with these 2, which is easy to do, and then kill them, and just get the one I know is going to work. So because it's easy to say, well, they should fund only risky research, but of course, a lot of that won't work. So you have to decide, if I have limited funds, am I going to go for the thing that will definitely work, but be incremental, or am I going to fund one which might make it get nothing? So in my view, we should stop funding projects. I can make up stories and write grants. I'm actually pretty good at getting grants, I should have to say. The NIH has been nice to me. I'm really great at making up stories, and whether I actually do what I say in there is really almost irrelevant, because once they give me the money, I'm not giving it back. So what I think they should really do, and I'm not the only one that thinks this, is you should bet on the jockey, not the horse. You should bet on the researcher, just give us some money and say, you can do anything you want, but in 4 years, I want to see what you got. And if you don't have anything, then we're going to drop your money. And if you have a lot, we're going to give you more, or we're going to raise it. So in other words, make it performance dependent and not sort of, how well can I tell a story?
Yeah, so invest in the person, don't invest in the project.
Exactly.
And they are going to move, and they are moving already in this direction. And you said that this may have been prompted by some of your input.
Well, I don't want to take credit for it, but we did a project with Francis Collins on diabetes, and we had this conversation. And I think this this conversation, and I guess I was also quoted this in an article on this very point. I think many of us realize that the R01 structure is broken. It was really meant, I do remember this, it was really meant that you're supposed to get one R01. It was supposed to be enough to run your lab. This was the days before molecular biology, when there was enough money there to pay you your poor little salary, which was really low, and the little bit of reagents you needed, which wasn't much. And then what happened is the whole enterprise blew up, and we need 4. I have 4 R01. We need 4 of them to run a lab. That's crazy.
Yeah.
Right. And so I think you just have to change it. And like everything, scientists tend to be a little bit slow to change things. But as you say, not just me, but many of us have come to them and said, look, you have to change that structure. It makes no sense.
We have Shri. Shri is the director of our nanomedicine center. And Shri just received a grant for undergraduate students also to have research, to do research with him, with Dana-Farber, with MGH, Brigham and Women, and maybe you at some point. So do you take undergraduate students?
Because we take graduate students? Oh, yeah.
Undergrad, because graduate and postdoc, we have that too. But we also, what excites me is that, we have many students involved in the labs.
Oh,
and
absolutely.
so can we send you some?
Sure.
Absolutely. All the undergraduate students, make sure you talk to him.
And I'm gonna put in an extra point to that, which is that we've really very much tried to rethink both what we do with the undergraduates and the graduate students. We've started what I think is the first in the country entrepreneurship track, design, technology, and entrepreneurship. We are completely rethinking undergraduate and graduate training. I mean, truly, if you want to talk about it, I can. I'm also associate dean at Sinai, you should know this. And so it's my job to do these sorts of things. And we're trying to rethink, do you really need the 5-6 year grind, or can we move through quickly and sort of keep that innovative spirit?
So can we stop here for a second? Why don't you expand on that? That's interesting.
Sure. So, okay, we're training people to be you and me, academic scientists. Now it's approaching 10%, down to 10% of people we train will be scientists. All right. So, and yet, unemployment rates. So look, everybody is panicking in the field. This is what a recession looks like. If you want to be a lawyer, it's worse. Okay. So let's have a little perspective here. Unemployment rate for a biology PhD is less than 3%. Okay? So that's not the issue. The issue is we're training for what they're not going into. So what do we do now? We've opened up a new entrepreneurship PhD, which is very non-traditional. For example, and it's run by a guy named Jeff Smith. Jeff is not a biologist. He doesn't have a PhD. He's a lawyer. But he ran a venture capital company, a successful one for many years. He invested in over 100 different companies. He's now a professor at Sinai, and he runs that entrepreneurship track. You can take a course called QED. Here's the course. First semester, learn how to start a company and start a company. Second semester, get the company moving, and then at the end of the second semester, you make a presentation to a series of VCs, and if you are one of the four best companies, you get money put into you, and you get an A. Okay? That's how you learn how to do it. And so for all the various, we're having a series of brainstorming sessions right now, which we're very big on, with various factions, including the students and the faculty and the chairmen. Each one, we're having 7 different brainstorming sessions. Everything gets thrown up on the wall. What exactly is the purpose here for training? I've set up a pipeline to JP Morgan, for example, and we have students that are now going straight into there. Why would JP Morgan want a biology PhD? What they tell me is the business is straightforward. The quant are straightforward. They can bring mathematicians in. But what they need is people who can think creatively but rigorously. And that's almost uniquely right down the middle of the biology PhD. They love them. Okay? So there really is a great world out there that you can leverage this. But we have to be more creative, and we have to look more clearly at what the world looks like, not what it was when I was growing up. Okay.
Great. Sri?
Yes. Rather than ask my question, let me actually follow up on the educational aspect of the grant that Joseph mentioned. I think your talk here inspired many of these students, and we want to, now we have an opportunity to provide them a research experience through this new CanCure program. So I hope that many of you will take up on this opportunity. There are flyers outside that you can look at, and you make the application through your co-op advisor. So I think it's great that your talk is going to inspire all these young students.
Yeah, that sounds amazing, and I think Sinai, I can say this, I think we would love to talk about partnering with you and seeing what we can do to leverage that.
That's great. So it's Dana-Farber, MIT, Harvard, Brigham and Women, MGH, and Mount Sinai. We just added. So the flyer is obsolete. We need a new flyer.
You need to remove most of the
So
others.
Sri, any question?
Well, yeah, I think one of the issues, I mean, and so I do test nanomedicines for cancer. And the current paradigm is, you do cell IC50s, and then you move into mouse models. And of course, the hottest thing now is patient avatar, the mouse avatars that you described. One of the issues with them is they have a long lead time to grow. So between the time you diagnose the cancer and you can get a treatment, you lost several months. So it seems to me your Drosophila is because of its much shorter life cycle, can play a role in that, and I'm sure you already know that, and that's why you're doing it. Am I correct in this?
Sure. So flies have a 9-day life cycle, which I love. And actually, you put your finger on a key point, which is a thing that worries. Excuse me, I think I'm hitting puberty again.
Yeah. Would you like some water?
Thank you. The thing that worries. I can't talk and yeah. No, I'm good. I'm good. Relax. The thing that worries me the most is actually the mouse model. So the flies will be accurate or not, and we won't. We'll find that out when we move through. I think we've taken them as far as we can go. But for example, I talked about the patient-derived xenograft, which I think you're referring to. I think there is a worry now, shall I say, that they may also fall down. So this is where you take a chunk of patient tissue, you put it in a mouse. It has all the complexity. The problem is, what happens actually in a mouse is it balls up, most of it dies. This crazy blood supply sort of leaks into it. You feed the mouse a drug, it all pours right onto this half-dead tumor. It looks great, but the problem is that clinical trials are now going poor, and I think Dave Tuvison has really been pushing this point. So now we're exploring other technologies, which I can talk about. So my point here is, I think there's going to have to be some real innovation on the mouse side to really have us leverage the fly side. It's actually the mouse part that worries me the most.
Okay, we have Dean Fulmer here. A question on this side, then a question on this side. Yes.
That was terrific. Thank you so much for your presentation. And you commented about the structure of your lab. Can you say a little bit about the role of philanthropy in the way that you support your work? If I know Ken Davis, you're probably pretty busy with that, but what proportion or what's your vision with that?
Yeah, that's a great question. So philanthropy is key for two reasons, okay? And of course, when I worked on basic development, philanthropy was really not even on my radar, okay? So I have to say that for me, this is a bit new. So it's important for two reasons. One is because, as you point out, the NIH budget is tightening, okay? But I think more interestingly, what I've really come to learn about and appreciate about philanthropy is that this is money that I don't have to convince somebody that it will definitely work. Because very often the people giving the philanthropy, they've gotten where they've gotten because they're people that like risk-taking and taking. I mean, how else would you get to that point in your life? In my experience is they've been people that like it when I say, this is going to be risky, it might not work. And all I have to do is convince one person, usually a pretty bright person, but just one person, that this makes sense, and then I can move forward with it. As opposed to going through a slow grind where I have a group of people, there's a little bit of groupthink going on. But as I said, at the end of the day, as we all know, I have to be very conservative about what I put in.
Did you say 20%?
No. Could you repeat that?
Sure. What percentage of my budget is philanthropy? Well, for the center, it's 100% because, as you point out, the NIH just had no idea what to do with this. I talked to them, and they were just lost. They said, that sounds great. We just don't have any structure for it. I think they're trying to change that, but not today. Okay.
Hi. I wanted to know a little bit more about the impact the center's had so far, how many people you've been able to treat, that kind of thing, and where you kind of see your projections going forward.
Sure. So where are we at? So we officially opened our doors September 1st. We've created a lot of fly avatars, and we're just beginning treatment of patients now. So I would say, I would just summarize by saying we don't really have data yet. And in fact, what I'm told I have to be careful to do is to not think too hard about it for probably the next 18 months. This is our goal. We want to get 50 patients for each of the tumor types. Because first of all, that's why we limit what we get, because otherwise we have like 5 of this and 7 of that. We need statistics for 2 reasons. One, I need to know if this is even a good idea. And I have to say the bar for colorectal cancer is so low that if we can't improve over that, we should be out of business. But secondly, we need insurance companies to pay for this. So let's think about this a second. A typical cancer drug can cost $10,000 or more per month. We come to a patient and say, here's your 3-drug cocktail. We can bankrupt them pretty fast. So far, foundations and philanthropy and so on and drug companies have been amazing at helping us out so that this doesn't happen, but that's not going to go on forever. So we need to show that this is better than standard of care so that we can make this a standard of care and so that people can actually afford it.
Hi. So earlier you said that before you were working as a scientist, you had started as a musician. And it seems that currently you're taking a very creative and unique approach to curing cancer. I'm wondering where there's overlap in your training. Personally, as an engineering major, I find that having a creative background and mindset is a very valuable asset. So I'm wondering if you apply any musical or artistic concepts from your past life, if you will, to your scientific work now.
Let's see. So I was obviously so successful as a musician that I became a scientist. And I had a band in Chicago, and I was pretty much a tyrant. And so what I learned from that was that I had to learn to listen to people because they weren't playing exactly the way I heard it in my head, because of course I wrote everything, because I had to be a control freak on that. But I know the question you're actually asking, which is the interface between science and art. And many years ago, someone wrote, it'll come to me eventually, I'm just blanking on his name, wrote a great piece where he argues that artists and scientists are actually much more similar than you would think. And because artists have this vision that scientists are these, like computers, so you just plug things in and then they magically put it out. But of course, that's not the case. The person who does the experiment, the way they do the experiment and the way they report the experiment very much colors the outcome of how people understand what has happened. Okay, I know it shouldn't be that way, we should all be computers, but we're not. Also, there is that creative aspect, which one of our goals in our training, you asked about that, is to not stomp that out, but to actually encourage this sort of thing of, doing brainstorming, having idea sessions and so on, and freeing people to not get lost in the doing so that they can get to some of the thinking as well. Ironically, and I really am getting to addressing the question, ironically, one of the things that I think is most important for young scientists, and I started a course called Becoming a Professional Scientist, okay? And it runs, and the next day I have something called Thinking Science. And the point of it, and they are the two sides of it. The Becoming a Professional Scientist, besides teaching leadership and so on, we teach basic organizational skills. Because if the doing gets in your way, and you guys know exactly what I'm talking about, or you will, if the doing gets in your way, you're not free to do thinking. You end up having to repeat experiments over and over, and it gets in your way. But if you can get that locked down, just like learning an instrument, okay, I play a number of them. And if I don't have to think about how to play a guitar, then I can dream out while I'm playing it and think about things, which is what I do, okay? It's the same thing in science. If you can get that under control and get that under some rigor, then you free yourself to begin to think about things and why you're doing it. This is where you want to be as a scientist. So in that sense, they're very similar.
Well, Ross, thank you very much for coming. And let me mention that since now we have an official partnership, it's on the web. I'm going to invite you to come and talk to Ross about anything. He will be here, and please feel free to mingle, ask your questions. But let's give him a big round of applause. Thank you for coming. Thank you.